I'm Stuart Reynolds, and I'm co-convener of the Science Group at the Bath Royal Literary and Scientific Institution, or BRLSI. In this short Virtual BRLSI video, I'm going to talk to my colleague at the University of Bath's Department of Biology and Biochemistry, Professor Jean van den Elsen, about some important scientific news that was announced just 2 days before our conversation took place. This was the announcement on November the 30th, 2020 by the London-based Google offshoot DeepMind, a company that specialises in artificial intelligence, or AI, that it had solved one of the most important problems in biological science today, the so-called protein folding problem. The idea is that it should be possible to predict the 3D structure of a protein from its one-dimensional amino acid sequence. Although the community of scientists working in this field has been confident for at least 50 years that this should be achievable, it's remained an aspiration. The announcement that the problem had been cracked was made at the biennial Protein Structure Prediction Challenge called CASP, short for Critical Assessment of Structure Prediction, and it's caused a scientific sensation, reaching the front pages of the world's media. I've chosen to talk about this here because this kind of interdisciplinary work is exactly the sort of area that appeals to the membership of the BRLSI Science Group in combining cutting-edge work on molecular biochemistry and computational artificial intelligence with implications for application in drug discovery and production and clinical practice. It represents a noticeable advance on what went before. Before we start, I'd like just to remind you that proteins are the components of living things that enable them to do things, like converting chemicals from one kind to another, sensing the external environment, recognizing self and non-self, capturing light, generating heat, light and movement. Not only that, but proteins are also variably shaped building blocks that are used to construct the bodies of living organisms, like Lego bricks, only more complicated in shape. They're able to do all those things because protein molecules have a bewildering variety of 3D forms. Their various protein shapes arise because when it's synthesized as a linear chain of connected amino acid building blocks, the way that the protein molecule folds determines its 3D shape. It's this unique shape that determines the functional properties of the protein. Starting in the 1950s, structural biochemists have worked out methods to determine what these shapes are in the laboratory, using X-ray crystallography, nuclear magnetic resonance spectrometry, and more recently high-resolution electron microscopy. But these techniques are all very slow, expensive, and there are literally millions of proteins for which we like to know their shape. And if we could only know their shape quickly, then we'd understand their function better and more quickly, and taking a medical point of view, it would enable us to interfere much more effectively with protein function for therapeutic reasons. As I'll discuss with Professor van Elsen in this interview, it's possible that solving the protein folding problem would greatly accelerate the production of vaccines for infectious diseases like COVID by enabling us to identify those parts of viral proteins that are vulnerable to the human immune system. Jean van den Elsen is a protein biochemist who's an expert in molecular structure determination. He works with protein components of the innate immune system, and he's also interested in vaccines and their thermal stability. All of these are subjects that qualify him very well to discuss these new findings with me. Neither of us have had any involvement with the work, and I hope that we're able to discuss its importance impartially. And I'm mentioning this because the new protein shape prediction software produced by DeepMind has the potential to make a lot of money. Moreover, unusual in this branch of science, it's been done by a commercial company rather than a university lab. I should also mention, of course, that this video was recorded at a time when both Jean and I were kept at home by the COVID pandemic. So, our conversation took place using Zoom. Good morning, Jean. It's very nice to see you. And I'm really pleased that you've agreed to talk to me about some hot news in the field of structural biochemistry, a subject that doesn't often get onto the front pages of the national press of pretty much every country in the world.
Certainly
And
in Corona times, yeah.
yeah. So, as I understand it, the news is basically this, that a company, so this is a commercial enterprise called DeepMind, which has set itself the task of predicting protein structures from the amino acid sequences of those proteins, thinks that it has come up with a breakthrough. I'll just pause long enough here to remind viewers of this video that proteins are long linear sequences of subunits of 20 different kinds called amino acids. And it's for a very long time, it's been an item of belief among structural biochemists that all the information that is necessary to define the three-dimensional structure of a protein is inherent in that sequence. So what does this new scientific finding achieve that we couldn't do before? And actually, do we believe it?
Yeah, so the DeepMind protein folding algorithm says that we now can predict how proteins fold in 3 dimensions without the need of experimental structural biology. So the methods that we use in the lab, such as X-ray crystallography or nuclear magnetic resonance, NMR, or cryo-electron microscopy. So these results are part of the biannual global competition called CASP, that's Critical Assessment of Protein Structure Prediction, which has been running since 1994. And this has become the gold standard for assessing predictive techniques. So yes, I think we should be very enthusiastic about this, because this being the gold standard for these predictions of protein structures, show that it's a really important result. So yes, I think we should believe this, and you're right about the fact that although information in molecules such as DNA and RNA and an amino acid sequences are two-dimensional, when a protein folds, it adopts a three-dimensional shape. And we always thought that by knowing the amino acid sequence, we should be able to predict this three-dimensional structure of a protein. But after, I think we've now solved over, not me, but in general, the structural field has solved over 170,000 protein structures. And it became clear that although for some amino acid sequences, it's fairly easy to predict how they fold up in three-dimensional space, but for others, it's been proven to be impossible. So with new algorithms and new software, new hardware, faster computers, it will be great if we can resolve this issue. And they've given this DeepMind computer this algorithm called AlphaFold access to a large number of structures that are available, over 170,000 structures, and we know that that although many of these structures are unpredictable based on their amino acid sequence, we know that there's only a limited number of folds that proteins can adopt. So there's a limited number of angles and distances between these subunits, these amino acids, that they can sample which means that a computer should be able to address these complex issues that we as humans cannot.
Yeah, yeah. Of course, inherent in what we're saying is the idea that a protein's function is defined by its shape. When you, in your laboratory, determine the shape of a function through X-ray crystallography, you're actually looking at the structure of the protein in a very particular circumstance, when it's very concentrated and in the presence of lots of other copies of the same protein. But actually, quite often, proteins aren't like that. They're free in solution. Or on the other hand, they might be embedded in other structures, like in membranes. I mean, is there anything that might be a problem there for the DeepMind algorithm? Are they looking
yes,
specifically at solution structures, or?
That's true. Well, I still need to have a look at the paper, because the paper hasn't been published yet. But I presume they sample from a wide range of different structures, including crystal structures, which, of course, because of the reasons that you mentioned, are static structures, because they get embedded. So you get many different protein molecules in a lattice, which is quite confined and can stop loops from moving. So you lose the dynamic information. And I think I totally agree with that. I think dynamic information is probably missing largely in the sample of structures they have. Although from solution structures, such as using techniques such as NMR or even cryo-EM to a certain extent, you can sample a larger range of these dynamic movements that proteins can sample.
You mentioned just now that the way that this prediction program works was through an artificial intelligence procedure, where the program was literally trained on a large data set of protein structures. Do you think that the proteins that we already know about are enough to train the DeepMind program to go beyond that range? Are there dangers in extrapolating to other kinds of proteins that are currently very difficult to determine?
I think you're making a really good point there, because there are proteins for which we haven't been able yet to solve the structure, although new structural determination methods have yielded a wide range of new protein structures that we were unable to resolve using NMR and crystallography. And you mentioned membrane proteins, which now, with the revolution of cryo-electron microscopy, now become part of a larger subset of proteins that we can now sample, and a DeepMind should be able to sample in the future. But I agree, there is some underrepresentation of these proteins, which we have yet been unable to solve their structures. And what I also know from, and this is from previous outcomes of this CASP meeting, this biannual meeting that these results have come out of, is that these algorithms tend to be very good at predicting protein structures that consist of helices. So amino acid strands that kind of wind up like a helix. And that might be also the reason why this algorithm is called AlphaFold. But proteins consist of other structural features as well. There are actually three main features: an alpha helix, but also long extended strands that amino acid sequences can adopt, and flexible loops that can connect helices and strands. So it could be that it might still prove difficult for proteins that consist of beta strands or combinations actually of strands, helices, and loops for DeepMind to be able to resolve such proteins and such structures.
Yeah, it's very encouraging that they've used this artificial intelligence approach, I think. I often liken this to a bit like learning to drive a car. You just get better as you practice more. And of course, the more structures that we know about, the better the algorithm will get.
Yes.
Yeah. Yeah.
So we know, yeah, we still need structural biology to be able to expand the data set.
Yeah.
But I guess there will be a point at some time that we have such a wide data set that we don't need any structural methods anymore to be able to resolve.
What will you do then, John? That's your job.
I'll be oh God, yes, I'll be out of a job. So I'm expecting an email very soon. But I have to say here that we have to look at protein structure also in a wider context. So in the context of biology, there are added levels of complexity. So proteins don't exist in isolation. They interact with substrates, ligands, and they interact with other proteins. So even within a single protein, there are multiple parts, which we call structural domains. So in one protein, you can have a structural feature which does one function, but the other end of the protein has a completely different function and folds differently. And to give you an example, I'm currently working on a structure of a protein called C5, which is important in the human immune system. And it consists of 12 of these individual domains. So whilst DeepMind algorithm, the AlphaFold algorithm, would be able to predict the structures of these individual domains, it's not yet able to predict how these domains interact with each other in a three-dimensional space. So how these parts can move with respect to each other, because proteins are not static, they are dynamic, as we've talked about. And then, of course, how do such proteins interact with partner proteins? How do proteins, for instance, on a cell surface interact with a receptor on another cell? So how does insulin interact with an insulin receptor? So in the context of biology, I think there are added levels of complexity for which we don't have any algorithms yet to predict how they interact.
I'm glad to hear it. It sounds as though there's plenty for you still to do.
Yeah, luckily.
So just to conclude now, what sorts of thing will the existence of this new predictive power enable people to do that they couldn't before? Maybe we could take an example of a vaccine. I know that you're interested in vaccines. So just as an example, would it have been possible that the development of a vaccine for COVID-19, would it have been easier or quicker if the DeepMind AlphaFold algorithm had been generally available to structural biochemists earlier this year?
Well, actually, earlier this year, DeepMind released predictions of structures of several of the SARS-CoV-2 proteins, so proteins from the COVID-19, so the coronavirus, I should say. And it released these structures before they actually had been determined experimentally using structural biology techniques. And it turned out that DeepMind's predictions for one of the proteins ended up to be very similar to the structure that was later determined using cryo-electron microscopy. So that is absolutely really impressive. I think. So I can clearly see practical applications here with respect to vaccine design. So you could imagine that with the outbreak of a new pandemic, we could use algorithms like this to predict structures of proteins on the surface of a virus or a bacterium really quickly, even before structural techniques can get to the structure. And then, for instance, we could produce some of these proteins exposed structural features, such as in the shape of short folded strands of amino acids that we call peptides. And we could use those folded peptides potentially to challenge our immune system and to make sure we're protected when we're actually exposed to the real pathogens. So I really see a potential practical use for this. And I understand there's a specific interest in also new treatments for parasitic diseases such as malaria. And I presume the reason for that is that nearly half of the world's population lives in areas that are at risk for malaria transmission. If you think about the estimated number of malaria deaths stood at over 400,000, I think, last year. So it's a serious global health problem. And there are other parasitic diseases that are considered neglected based on their high prevalence and strong links with poverty. So I could see using an algorithm like that to come up with potential treatments or vaccines really quickly.
Oh, that's a very good point, Jean. Well, I noticed that in the newspaper that I read the story in, that a well-known structural biochemist had said, this changes everything. And well, I think that perhaps that's a bit of an exaggeration, because you've shown us this morning that the themes that people were already thinking about will carry on being of interest. So it isn't that this really changes everything, but maybe it speeds it up a lot. So thanks very much for talking to us this morning, Jean. BRLSI is very grateful to you. You're a good friend to us, and I hope that I'll have opportunities in the future to talk to you again. Thanks a lot.
Yeah,
Bye.
thanks very much, Stuart, for inviting me. Yeah, it's becoming pretty clear that there's still a lot of work to do, but we can definitely conclude that the future folds rather than unfolds.
That's great. Thanks a lot. Goodbye, Jean.
Goodbye, Stuart.